It's a well-known protein or growth factor that's expressed in different tissues of the body, part of the normal immune system and regulates the function of different cells. And among other things, it's known to be widely expressed in the liver and in hepatocellular carcinoma. And part of the issue or role there is that TGF-beta is known to have very potent immune suppressive activity. So it binds to certain receptors on different immune cells, T cells, natural killer or NK cells, and so on, and actually inhibits their activity. And so it's been shown in other studies that, basically levels of TGF-beta expression in HCC is a negative prognostic factor and also may, contribute to the development of HCC itself. It's known that different immune cells have receptors for TGF-beta, and among other things, this inhibits their activity. And so this is probably part of the reason why HCC can develop. It's sort of, it's part of the so-called immunosuppressive tumor microenvironment, which this is not the only factor. There's a number of cells and proteins and such that are expressed in different tumor types that can promote tumor growth in part by preventing what would be the normal, body's reaction against the tumors. So in part, this production of TGF-beta by HCC likely limits the normal, immune response and allows for tumor to develop. And then also, a number of groups have been testing different immunotherapies against HCC, with relatively limited efficacy. And, we sort of hypothesize in part this is due to TGF-beta. And if we knock out the TGF-beta receptor on our immune cells, in particular, we're interested in natural killer cells, this could improve the anti-tumor activity, which is essentially just what we saw. The interest and kind of expertise of my group is production of natural killer cells from human induced pluripotent stem cells. So these iPS-derived NK cells have the normal sort of phenotype and function of so-called regular NK cells or NK cells that might be in your body. And among other things, they're known to kill certain tumors and virally infected cells. But other tumors are more resistant, including HCC. And so our group and others have expressed CARs, chimeric antigen receptors, on iPS-derived NK cells and other NK cells and different T cells and so on. But we can express CARs on the NK cells. And then one of the advantages of working with iPS cells is it's relatively straightforward to knock down or inhibit expression of genes of interest in the cells, because we can do all the engineering in the iPS cells and then derive the NK cells that are all homogeneous for that knockout. And so what we did here was compare either adding a CAR that targets HCC cells. So we tested two different CARs against either GPC3 or alpha-fetoprotein, and other groups have used, anti-GPC3, at least CAR T cells for HCC, I think with relatively modest results. And then we also used two different strategies to inhibit TGF-beta mediated activity, either by knockout, complete deletion of the TGF-beta receptor using what's now a pretty standard CRISPR-Cas9 system, or we expressed a dominant negative receptor for TGF-beta, which again, other groups have shown inhibits the activity. And so we did really either one strategy or the other or both. And what we found was really pretty clear. If we add, so normal, so we tested a couple different HCC cell lines that are resistant to unmodified NK cells. If we added a CAR to those cells, it really didn't do much. And we demonstrated this CAR worked on other non-HCC cells. So the CAR works, but when, testing against these targets, it really didn't do much. When we knocked out the TGF-β receptor expressed the dominant negative receptor, the NK cells actually killed the HCC cells quite well. Killed them in vitro, we test them in vivo, we get prolonged survival of the mice. And when we combine then the anti-HCC CAR with the TGF-β knockout, the CARs didn't actually add that much to the benefit. And so really it shows the both requirement and I think potency of knocking out the TGF-β receptor or inhibiting that immune suppressive activity in NK cells to mediate their anti-tumor effects. Now, NK cells are different than T cells because they have many other activating and inhibitory ligands that regulate their anti-tumor activity. So NK cells express proteins like NKG2D and so on that recognize certain ligands on tumor cells, and we could express these, or we could show that these were expressed on the NK cells in the tumors. So essentially, by knocking out or preventing TGF-β mediated inhibition, we kind of unleash the normal anti-tumor activity of the NK cells. And so, I think this is important for clinical translation now of all these groups, testing different cell-based therapies against HCC and other solid tumors. We just happened to test HCC because we knew it made a lot of TGF-β. It's a very challenging tumor, but there are many other tumors that express lots of TGF-β as well, ovarian cancer, pancreatic cancer, so on. And so the results are probably broader than what we tested, and it's probably true for T cells as well. So again, all those CAR T cell projects or trials that show relatively modest efficacy, if you can inhibit the TGF-β mediated effects, it's probably going to boost their activity there. I think if you're trying to target a tumor that's known to produce lots of TGF-β, like those that I mentioned, then yes, it's probably going to be beneficial. And I think this is something that we want to emphasize to all those different groups and companies that are testing these different cell-based therapies. Well, the challenges, I think have been relatively modest anti-tumor activity for most solid tumors, whether you're using, different autologous or allogeneic CAR T cells or NK cells, which are typically allogeneic. Again, I think there's a lot of literature about this, again, about the so-called immunosuppressive tumor microenvironment, that there are many factors that have to do with solid tumors that make them more challenging than hematologic malignancies, where we've seen much greater benefit of, especially CAR T cells, right, which for CD19 expressing B cell malignancies, have been really a transformational therapy, similar in multiple myeloma now, using different CAR T cells, really big effect. So solid tumors have issues, again, of more immunosuppressive factors, of homing to the tumor. You might have a limited, area that they need to get to. They have to penetrate the tumor, and again, a number of other factors that make these challenges. So TGF-β is one of these, right? I don't think this completely solves the problem of or challenges of treating solid tumors with CAR T cells or NK cells, but I think it provides a key piece of the puzzle. Well, we hope that this will be translated into a clinical therapy, either directly or indirectly. Again, I've worked with different companies developing iPS-derived CAR NK cells. There's certainly many more companies developing CAR T cells of different shapes, as well as other NK products. And I think, that as the field continues to advance, people look at this as something to add to their engineered product. There's many trials going on now. So I think, a lot of companies sort of have their pipeline, but, hopefully this is something that will be added to the pipeline down the road and something we can get into patients to improve outcomes for this type of tumor.